Hello people, welcome to my channel. I'm Nandini. So today I'm going to discuss about the overview of genetic engineering and its techniques to how to cure genetic disorders using gene therapies and many other techniques. So without much delay, let's start this video. So basically genetic engineering is a vast subject and it has become a great research topic in recent years, right? So let us discuss in a brief manner about this topic now. So in this slide, let's discuss about the gene therapy. So gene therapies can work by several mechanisms, right? So upon the several mechanisms, the main technique or the main process is it modifies a person's genes to treat or cure disease. So it can work by several mechanisms like replacing a disease causing gene with a healthy copy of the gene or inactivating a disease causing gene. It is something like suppressing the disease causing gene and modifying it with the insertion of a new gene from the other organism. So now, upon this research, upon many researchers, genetic therapies are approaches that treat genetic disorders by providing the new DNA to certain cells or correcting the DNA. So there are many gene transfer approaches. So usually a gene is transferred from one organism or from one part, like from one part or to the other part through using a vector, which is a major like a major advantage in the genetic engineering. Vectors are those which act as vehicles. So these will actually carry the gene from one organism to the other organism or like host cell, which is a major basic steps for gene cloning and etc. So vectors are again many types. It is again a big topic which is something different from mine, like plasmids, cosmids, phage and etc. So there are many genetic disorders which can be cured and which cannot be cured. So upon genetic disorders, as I have discussed before in the previous slide, that like thalassemia, genetic disorder, and many thyroid problems like hypothyroidism and phenylketonuria and etc. Many other genetic disorders have some sort of techniques to be cured, but not completely. It can be prevented rather than cured. As we all know that prevention is better than cure, gene therapies can be suggested or prescribed to cure or to prevent such disorders. Diagram, we can understand a clear-cut examination or clear-cut techniques of the gene of this gene therapy. So the first process will be cells from the patient or cells from a deceased diagnosed patient will be removed, and the second step would be the laboratory. As I told you before, that it is an ex vivo process. Ex vivo is nothing but the modification that is taking place outside the body. So in the laboratory, a virus is altered, like a viral vector is altered so that it cannot reproduce. Alteration is nothing but it cannot reproduce or it cannot clone itself. So the third step would be a gene is inserted into the virus. In this viral vector, the gene which we are selected from the patient can be introduced. As I told you before, vectors will act as vehicles that carries the genes from one organism to the other organism like host cell. And the next step would be the altered virus is mixed with the cells from the patient. Now this altered virus that have no capability to reproduce will now mix with the cells of the patient. As the patient DNA or the patient cells are removed from that, they both can be mixed together. And now from this, the process is carried out. Now the cells from the patient become genetically altered. Not, it is nothing but genetic alteration is nothing but now the cells which are already affected, we'll be collecting the cells which are already diseased or already affected, and now with this viral altered gene, as we are mixing this viral altered gene with the normal cells, now upon this mixing, the normal cells of the patient or like the disease cells of this patient will also get altered. They are genetically altered, and hence after this genetic altered cells introduction into the patient, now the disease can be cured or it can be prevented because of this alteration, and there would be no reproduction of the cells. It is a major advantage for cancer. This process is usually done majorly to prevent or to cure cancers, many types of cancers like that. Last but not the least, rare genetic disorders are those which are very rare in the world, like only upon, if there are 100% of the population, in 100% of the population only 2 to 3% of people will be affected with such genetic disorders. Upon these, which I have explored, they are fibrodysplasia progressiva ossificans or fibrodysplasia ossificans progressiva, which is considered as FOP in short form, which is the formation of bones or the extracellular, extraskeletal bones on the soft connective tissues, like the whole body. Upon years, upon months, it will become as stone, like the bones will be growing extracellularly, which is really a hard situation for a person to move his or her hand. So it is considered as a rare genetic disorder, as I have mentioned here, that there are few other diseases like mitochondrial diseases, adrenoleukodystrophy. Majorly, these rare genetic disorders are usually restricted to muscles, bones, and etc. So, upon many researchers and upon many scientists, it was known that gene therapy techniques are yet to discover or yet to cure such rare genetic disorders, and it is really a great challenging task for scientists or for any other person to identify such gene mutations and to cure such diseases. And hence it is becoming a great difficulty task to cure such diseases and to cure such genetic disorders. I hope you like my video. Please